By the 1920s, biologists knew the basic address of inheritance. By peering into cells, they had tracked genetic traits right down to the nucleus, specifically inside the chromosomes. But the exact chemical running the show inside those structures remained completely unknown. If you analyzed a chromosome chemically, you would find it was built from two primary ingredients, DNA and proteins. At the time, the scientific consensus heavily favored proteins. Proteins are intricate, built from 20 different amino acids, which gave them the structural complexity seemingly necessary to store the vast code of life. DNA, by contrast, relies on just 4 basic building blocks. Most researchers wrote it off as a rigid, simple molecule, essentially functioning as biological scaffolding. Because the belief in proteins was so deeply entrenched, anyone claiming otherwise faced a massive hurdle. They had to bury the protein theory under a mountain of undeniable physical evidence. The first real challenge to the protein consensus came in 1928 from a bacteriologist named Frederick Griffith. He was studying Streptococcus pneumoniae, working with 2 distinct strains of the bacteria, one highly lethal and another completely harmless. This diagram shows the strange paradox of his experiment. Griffith combined lethal bacteria that he had intentionally killed with heat together with the live, harmless strain. He injected this mixture expecting the mouse to survive, but the combination proved fatal. When Griffith examined the mice, he found living, lethal bacteria. The dead strain had somehow passed its deadly traits directly to the harmless strain, physically transforming them into killers. Griffith called this mysterious transfer the transforming principle. His work proved that genetic information is robust enough to survive the deck of a cell and dictate instructions to a living organism, even if the actual molecule doing the work remained a mystery. It took another decade for a team led by Oswald Avery, Colin MacLeod, and Macklin McCarty to chemically isolate Griffith's ghost molecule. This diagram illustrates their methodical process of elimination. They used highly specific enzymes, essentially microscopic chemical scissors, to systematically destroy one molecule at a time in the heat-killed bacteria. When they destroyed the proteins, and then the RNA, the harmless bacteria still transformed into the lethal strain. But look at this final test tube. When they introduced DNA, an enzyme that specifically digests DNA, the transformation entirely stopped. The harmless bacteria remained harmless. By process of elimination, the team concluded that DNA, not protein, had to be the active hereditary material carrying the instructions. Despite the clarity of the chemistry, many biologists simply refused to accept the result. Skeptics argued the DNA samples might have been contaminated with trace amounts of highly active proteins. They demanded hard, mechanical proof before letting go of their preferred theory. That absolute mechanical proof finally arrived in 1952, driven by researchers Alfred Hershey and Martha Chase. They chose to work with bacteriophages. These are specialized viruses that infect bacteria by acting like microscopic syringes, latching on to the outer cell wall and injecting their genetic instructions inside. Because phages are essentially just DNA wrapped in a protein shell, they offered a perfect test case. The researchers grew one batch of viruses in radioactive sulfur, which uniquely tags proteins. They grew a second batch in radioactive phosphorus, which uniquely tags DNA. After letting the tagged viruses attach to the bacteria, Hershey and Chase needed to violently separate the outer viral coats from the infected cells. To do this, they threw the samples into an ordinary household kitchen blender, shearing the empty viral shells right off the bacteria. When they spun the mixture down to separate the heavier cells from the liquid, the results were clear. The radioactive protein was left floating uselessly on the outside. Only the radioactive DNA had actually entered the bacterial cells. The protein coats never breached the cell walls. By physically tracking exactly what material entered the bacteria, the blender experiment supplied the indisputable proof required to crown DNA as the master molecule of inheritance. With DNA confirmed as the genetic vault for most life, a secondary question emerges. Why does biology heavily favor DNA over its similar cousin, RNA? After all, some biological entities, like the tobacco mosaic virus, responsible for infecting this leaf, rely entirely on RNA to carry their genetic code. This compares DNA and RNA backbones. Look closely at RNA's tiny addition, a 2' hydroxyl group on every nucleotide. That reactive oxygen creates vulnerability, making the strand fragile. Under stress, RNA degrades and mutates quickly, while DNA holds its shape. Because DNA completely lacks this reactive oxygen group and swaps out the chemical base uracil for a much sturdier one called thymine, it operates as a chemically inert, heavily armored structure. To function effectively as the blueprint for an organism, a molecule must prioritize long-term chemical and structural stability. It needs to copy its code accurately, generation after generation, without degrading. In biology, structure dictates function. RNA's instability makes it dynamic, perfect for swiftly carrying instructions to build temporary proteins or power short-lived viruses. But DNA's rigid, unyielding armor allows it to secure complex genetic information permanently, locking down the code without misfiring or falling apart. When Griffith first heated those lethal bacteria in 1928, the DNA inside survived the fire. The 24-year scientific pursuit that followed ultimately identified a microscopic vault capable of withstanding extreme physical stress, the passage of time, and the grueling demands of evolution.